A graticule and a micrometer scale can be used to measure the size of biological structure that are viewed with microscope. So here we have a microscope structure, and number one is eyepiece. This is eyepiece lens of microscope where we can put graticule scale. And number two is an objective lens. That's a second lens in a microscope. And number three is a stage of micrometer where we are placing micrometer. So here the question about which row show the correct locations of the placement of graticule and micrometer scale on the microscope shown there. So we can see that the graticule we are placing in eyepiece, that's number one. And the location of the micrometer scale is the number 3. So the B is our correct option here. So the next question is regarding cellular organelles. So 6 organelles found in the eukaryotic cell, they are shown there. Number 1 is nucleus, and the function of nucleus is to synthesize messenger RNA. Number 2 is mitochondria, and this is powerhouse of the cell. It provides energy for making protein. Number 3 is our centrioles, and their function is to divide chromosome during cell divisions. So number 4 is endoplasmic reticulum. So this is our rough endoplasmic reticulum, which combined amino acid to produce protein. Number 5, we have chloroplasts. That's another double membrane bounded organelle, and their function is to absorb sunlight and produce the glucose, which in turn convert into starch, and their function is to make starch and to store starch. And number 6, we have Golgi bodies or Golgi apparatus. So these are stacks of membranes network, and their function is to modify protein, mean they combine carbohydrate with a protein to produce glycoprotein. So next we have which organelles are involved in synthesizing and secreting the glycoprotein. So the protein production process is involved. First, the nucleus, which contains the DNA, the genetic information, and this will produce messenger RNA. And this messenger RNA moves from nucleus to the cytoplasm, and it combined with the ribosome on a rough endoplasmic reticulum and produce protein. And this protein will proceed to the Golgi apparatus and combine with a carbohydrate and convert into different products like enzymes or other products. So what organelles are involved? Number 1, the nucleus is involved. Number 4, the endoplasmic reticulum is involved. And number 6 is the Golgi bodies is involved. What about mitochondria is providing the energy for whole this process? So our correct option will be B, which contain 1, 2, 4, and 6. Which cell structure can have messenger RNA inside them? So cell structure like chloroplast and mitochondria, they contain circular DNA, and their circular DNA transcribes into messenger RNA, and they are producing their own protein. While the nucleus contains DNA, and this DNA is present in a chromosome, which transcribes into messenger RNA for production of the protein. While the rough endoplasmic reticulum did not contain messenger RNA. It produced the protein, and protein moved into the rough endoplasmic reticulum. So there's no messenger RNA in rough endoplasmic reticulum. So our correct option will be 1, 2, and 3. That is B. The next question is regarding the centrifugation technique. The scientists carried out an experiment to separate the organelles in animal cell by mass. So the scientists mixed the cell with the buffer solution, and they had same water potential as the cell, meaning it will not damage the cellular organelles. So cells were broken up with the blender, and then we perform centrifugation. Then after the centrifugation, we let the cell to settle down in the bottom. So the cellular organelles who settle down in the bottom, that was the heaviest part. And that made the pellet 1. So we remove this pellet 1, and then we remove the liquid above the pellet 1, and we transfer another test tube, and we did once again the centrifugation, and we let them to settle down in the bottom. So next heaviest particle settled down, and then once again we repeated this process for a third and fourth time, and then we got the heaviest particle again and again. And in this way, what is the answer for this question? So we need to understand the size of these particle and the particle which are heavier. So heaviest particle present in the animal cell is nucleus. So that has a high size. You can see 5 to 20 micrometer. And the next heaviest particle is a double membrane bounded organelle. That's mitochondria with a size 0.5 to the 1 micrometer. Third heaviest particle is lysosome with a 0.1 to 1.2 micrometer. And the fourth heaviest particle is there's no membrane on its surface, but it's a compact structure. And that's with a 20 to 30 nanometer. So these particles, they are designed on the basis of size. So you can see if the size is decreasing, the mass is also decreasing. So in this way, the pellet 1 will be the nucleus, and pellet 2 will be mitochondria, and pellet 3 is a lysosome, and in the same way, there is ribosomes. Now the question is about what is the main function of the type of organelles extracted in pellet 2? So the function of mitochondria is to production of ATP energy for the cell. So it is called the powerhouse of the cell. So in this way, the B is our correct answer here, production of ATP energy. So, and this type of question, examiner can also ask you about pellet 1 and pellet 4. So pellet 1 has a function, so controlling the cellular in all activities of the cell. And pellet 3 function is to have intracellular or extracellular digestion, while it contains hydrolytic enzymes. Pellet 4 contain the ribosome, and this ribosome has a functions to make protein. Which structures are found in palisade mesophyll cell and photosynthesizing prokaryotic cell? So this is question the structures which are present in eukaryotic plant cell and the prokaryotic bacterial cell. So the cell surface membrane that's present in both, and cellulose cell wall. So the cellulose that's present only in the plant cell wall, while bacteria contain peptidoglycan, mean this is protein. So that's not present in both. So ribosome, that's present in both for making protein, while chloroplast that's present only in eukaryotic. Why? Because it's a double membrane bounded organelles. And in the prokaryotic cell, there's no membrane bounded organelle is present. So that's why our correct option will be 1 and 3, that is C. Which polymer are present in all viruses and in all prokaryotes? So polymers mean when so many different monomers or small units link together and they produce polymer. So polynucleotide, when so many different nucleotide, for example, we have the nucleotide, adenine, guanine, you studied about adenine, guanine, cytosine, and thymine, different nitrogenous bases. They are combining with the ribose, sugar, and phosphate and making nucleotide. These nucleotides link together and produce the DNA or RNA. And these are polynucleotide. So both viruses and the bacteria, they contain the nucleotide. Polynucleotide, like some bacteria and bacteria contain RNA and DNA both, while the viruses some viruses contain RNA, some contain DNA, but that's a common character which is present in both. We have polypeptide, it's protein. So protein is present in both. The bacteria contain mostly the protein in the form of peptide or glycan in the cell wall of the bacteria, while the virus contain the capsid or small capsomere protein. They join together and making outer coat of virus. So that's why the virus also contain protein, while the polysaccharides like starch and cellulose and glycogen and lignin, these type of the polysaccharide molecule, these are not present in most of viruses. So that's why that's our 1 and 2 that present in both. So B will be our correct answer here. Now, question number 7 is related with emulsion test of lipids. So first we can see that in the down in the bottom, you can see the emulsion test of the lipid. So emulsification test is first, the step number 1, we mix the sample with ethanol and shake it gently. So ethanol is it can dissolve the lipid. So lipid can dissolve in ethanol. So after dissolutions, so then the second step, we add water with that. And after adding water, once again, when we will shake it, so we can see the small droplet of the lipids that will produce, and that produce a cloudy appearance. So if it is producing cloudy appearance, so it means the lipid is present. That's a positive result. But if it stays same, there's no cloudy appearance, so it means the lipid is absent. So that's the procedure for performing emulsion test. So A is an option. So add the 2 centimeter cube of water to the sample and pour the water in a test tube containing 2 centimeter cube of ethanol. And there is no shaking is involved in that. So that's why without shaking, it will not produce the cloudy appearance. So that's why A is our wrong answer. So B, add the 2 centimeter cube of ethanol and shake the sample, and that will produce the cloudy, that appearance will start coming. And then if you boil it, if you add water and shake, and that will produce cloudy appearance. But when you will boil it, this cloudy appearance will remove. So endpoint will be not clear. So that's why if there's a boiling is there, that's a wrong option. So in this way, B will be a wrong option. So C, add the 2 centimeter cube of water, and then once again, shake the sample, and then once again, there's a boiling is involved here. So by the boiling, emulsion will remove, and the solution will become clear once again. So that's why that's a wrong option. So correct option is coherent with the given procedure. First, we're going to add the 2 centimeter cube of ethanol to the sample and shake it. And then we pour the ethanol in a test tube containing 2 centimeter cube of water, and then shake it again. And the lipids are present, and then the mixture will become cloudy. So this is a correct method to perform emulsion test. So these are correct option here. A student was provided with a solution of carbohydrate. They remove 2 samples from the solution and perform the test on each sample. So you can see we have a carbohydrate solution, and we got a sample 1, and in this sample 1, we did Benedict solution test, and result came negative. So it means this is non-reducing sugar. So one thing is confirmed, like it is carbohydrate. But this carbohydrate is non-reducing sugar. It may be sucrose, or it may be any polysaccharide, which are non-reducing. So it's not a specific test for sucrose. We cannot say it is confirmed sucrose. We can say maybe sucrose or maybe polysaccharide. Next, we have a sample 2. In a sample 2, we will add dilute hydrochloric acid. Why? Because hydrochloric acid can break down the glycosidic bond between the carbohydrate, and this will reduce monosaccharide, mean reducing sugar. So then we will neutralize the acid with a sodium hydroxide strong base. Why? Because the Benedict solution is not working in the presence of acid. Then we will use this Benedict solutions and we will add the Benedict solution, and the result will be positive. So it means that is reducing sugar. So this whole procedure, and that's the part of also the paper 3 practical, we're going to see the type of questions might possible relevant to this. Which statement explain the results? So the number one, the condensation reaction occurs in the sample to release the reducing sugar in the result of the adding hydrochloric acid. So hydrochloric acid is breaking the glycosidic bond. So that's why this is hydrolysis reaction. It's not condensation reaction. Number B option, the glycosidic bond in a polysaccharide has been broken down to release reducing sugar. It is correct option. Why? Because when we add the hydrochloric acid, then it breaks the glycosidic bond and it's releasing reducing sugar. So number C, the sample on sample 1 showed that the sucrose is present in a carbohydrate solution. Now you can see the sample 1 is giving negative result with a Benedict solution, but it's not mean like this is a confirm that this is sucrose. It may be sucrose or it may be polysaccharide because they are non-reducing sugar. And the option D, there's a change in the color to yellow solution that shows the glucose is present. Fine, if there's the solution is turning to yellow, that shows the glucose is present, but it's not confirmed that the glucose is only glucose is there because so many monosaccharide, for example, we have glucose, we have gallose, mannose, and there are other fructose. So these are, they are also giving results with Benedict solution. And in the same way, like pentose sugar, that's also giving result with the Benedict solution. So that's why it's not confirmed like this is glucose. It's maybe any other monosaccharide, maybe maltose. So that's why this is wrong option. So our correct option is here, B. We're going to proceed to the next question. Is related with biological molecule. Which molecule contain at least 2 double bond? So we have sucrose, this is carbohydrate, disaccharide, and we have collagen, this is our fibrous protein, and hemoglobin, this is globular protein. So if you see that the structure of a sucrose is consist of the alpha glucose and beta fructose by 1,2 glycosidic linkage. And in this structure, if you're observing, there's no double bond is present. So that's why A, C, and D are excluded. Why? Because there's no double bond is present in the sucrose. While in the hemoglobin and the collagen protein, you can see there's the structure, and in this structure, there is the amino acid. So when the different 2 amino acids are linking together by the peptidic bond, so in the amino acid, you can see the carboxyl group is present, and this carboxyl group contain double bond. So that's why the B, which is shared between hemoglobin and collagen, this contain double bond. So B is our correct option here. We're going to move to the next question, is also from biological molecule. What describes the cellulose? So what is the description of cellulose? So cellulose consists of beta glucose with the linking of 1,4 glycosidic linkage, and every next molecule has 180-degree alterations. So that's overall description. What about these long chain of a long molecule of the beta glucose, they have no branch. So option 1, a branch structure and alpha glucose, this is wrong, while the cellulose contain beta glucose and there's no branch are present between the molecule. Second, a branch chain, so that's also wrong. And the C is option, alpha glucose is wrong in that. And the D option, an unreactive linear chain of the 1,4 beta glucose. So that's the D is our correct option here. Which part of the structure of a hemoglobin carries oxygen? So if you see that the hemoglobin is consist of the 4 polypeptide chain with heme group. So these 4 polypeptide chain cannot carry the oxygen, but they're carrying the carbon dioxide. For example, hemoglobin they're making, and they can carry the carbon dioxide, but they cannot carry oxygen. While heme group, this contain iron atoms, and this iron in the ionic state combine with the oxygen and produce iron oxide, and this can carry the oxygen. So that's why this is the heme group can easily carry the oxygen. So the B is our correct option. What about hydrogen? That's only the involved for maintaining the structure of protein. Hydrophilic R group. So these are hydrophilic R groups. Some contain positive charge, some contain negative charge. It facilitates the heme group to carry oxygen from water. So it provides only the facilitations due to the presence of charge. Why? Because it is making a hydrogen bond with the water. It can carry the water layer. So in this way, what is the oxygen is dissolved in the water, it can absorb into the hemoglobin. So that's why the B is our correct option there. So what is the maximum number of hydrogen bond that can form between two single water molecules? So hydrogen bond is what? There's a force of attraction between partially positive hydrogen atom of one water molecule and a partially negative oxygen atom of another water molecule. So what about there's a force of repulsion between partially positive N and negative N of one water molecule. So that's why there is only one hydrogen bond is possible between two water molecules. So if there's only two water molecules are there and only one hydrogen bond is possible between partially negative oxygen and partially positive hydrogen atom. So A is our correct option here. CYP3A4 is an important enzyme in human digestive system where it is needed to break down the range of different toxin. So I mean this enzyme is working detoxification, so it's breaking down the toxin in digestive system. So the activity of CYP3A4 has been shown there to reduce the substance called furanocoumarine. This furanocoumarine is found in some fruits, and so it's a dangerous concentration of the toxin may develop in a human digestive system when the fruits containing furanocoumarine are eaten. So in this way, what is they're showing? And they're showing like furanocoumarine is a chemical which is working as the irreversible inhibitor of enzyme. Now, enzyme is breaking down the toxin. What about this chemical? It's reducing the activity of enzyme. Now, question, from the information provided, what can be concluded about the molecule of enzyme? So here, simply examiner asking question, what is the function of enzyme CYP3A4? So that's a general question, but sometimes they're very simple type of the questions, but so they are molded by using a different word to make it more complicated. So simple is that what is the function of enzyme CYP3A4, or what's the function of enzyme? So function of enzyme is to lower the activation energy of toxin broken down reactions. So the toxin is broken down and for the breaking down of the toxin, there is need of activation energy. So the function of enzyme to decrease the activation energy and by using up low energy convert the reactant into the product. So while this function is reduced by that's our furanocoumarine and the chemical. So A is our correct option here. B is the wrong option. Why? Because they bind to the specifically through the active site to the substrate found in some fruits. So that's not given there in the question statement. So the C option, that's changed permanently when it acted by the furanocoumarine molecule. So you can see this is a reversible inhibitor. That's not given there, like it's permanently cancel the action or it's denatured. This is not given there in the question statement. It's not given there. It reduces the substance. So in this way, what is it? So that's our permanently change. That's not given there, this option in the question statement. What about in D? They resume the normal activity when the concentration of the furanocoumarine decreases. So it's not given there. So for example, if we are decreasing the concentration of the inhibitor, so the normal activity can be resumed. So what about that's maybe the correct options for, but this is not correct according to the given information in the question number 13 statement. So this way, whenever this type of the questions come, you need to focus about what the examiner is saying in the question, try to understand that statement and paraphrase in your own word and proceed for the answers. So we're going to proceed to the next question here. Fixed volume and the concentration of the substrate and enzyme were mixed, and all other variables were kept constant. So enzyme catalyzed reactions was left until it was complete. So you can see, number one, there's a fixed volume and concentration of the substrate and enzymes. So if there is a fixed volume of a concentration of substrate and enzyme, then this quickly enzyme will convert the substrate into the product. No further substrate is available, and the reaction will quickly decline. So you can see the C is our correct option here. Why? Because the substrate is limited, and enzyme is not increasing, and quickly the enzyme will convert the substrate into the product. And in this way, no more substrate is available, so rate of reaction will decline to 0. So we can see other options. A graph is possible when the enzyme is increasing with infinite concentration of the substrate. For example, if there's along x-axis, if there is enzyme and the substrate is infinite, then A is possible. What about the B? If you put the substrate along x-axis and it's increasing with a fixed concentration of enzyme. So if the concentration of enzyme is fixed and the substrate you're increasing along x-axis, so B option will be correct here. What about the D option? If you put the temperature along x-axis, and if you're increasing the temperature, when it will reach to the maximum, the temperature, or you can say optimum temperature, so rate of reactions will be maximum, and then it will decline. So that's why the C is our correct option here. So we have done, we're going to proceed to the next question. Which molecule in cell surface membrane are typically involved in cell recognitions? So cell recognitions mean that's any molecule which is present on the surface of cell surface membrane. For example, carbohydrate, they're linking with the lipid, and they are producing glycolipids. When the carbohydrate are bounding with the protein on the surface of membrane and producing glycoprotein. So, what about the some molecules which are present inside, or they're embedded inside of the plasma membrane? They're not involved in recognitions of the cell. So in this way, we can see that cholesterol is not involved in cell recognitions, while the glycolipid, the carbohydrate bind with the lipids on the cell surface membrane, and this is that's performing the role of cell fingerprinting. So that's also involved. What about glycoprotein? It's also performing its role. So that's for cell recognitions, while phospholipids. So these phospholipids are not performing their role. Why? Because they are common in all cells. So all cells, they contain same chemical property of the phospholipids. So that's why this is not a differentiating character. So cell recognition is more important. You studied about the immunity system. Our immunity system is working as a security mechanism inside of our cell. Any cell who is from outside of our body. So if you, for example, if we transplant the organs from outside to inside of our body, if we transplant any organ or any extra cell is coming inside of our body, or our cell is not behaving properly, so these cellular, the glycolipids and glycoprotein, they start changing their chemical nature. So our immunity system can easily identify and kill them. So that's why that is an important process, especially the removing cancer from the cell and immunity action against the different bacteria. So that's more important. what can increase the fluidity of cell surface membrane? So cell surface membrane fluidity can be increased by using double bond, so between the fatty acid chain of the phospholipids. But here option 1 is saying single bond between a carbon-carbon atom, so that is decreasing. So what about double bond is responsible to increase the cell fluidity? While the cholesterol, so this cholesterol is able to maintain the structure of a protein at high temperature and at the low temperature. So function of the cholesterol in a eukaryotic cell is to maintain fluidity in the cell, so cell membrane. So number 3, longer chain fatty acid. So longer chain fatty acid is an opposite. That's shorter chain fatty acid is increasing the fluidity. So in this way, that's the correct option will be 2 here. And what about option 1 and 3 are wrong? So we're going to proceed to the next question. The 3 main factors that affect the rate of diffusion across the membrane can be expressed by the relationship is given there. For example, the diffusion rate is directly proportional to the surface area, concentration, and difference, while inversely proportional to the thickness of the membrane. So you can see the option number 1, the surface area has doubled. So if you're doubling the surface area, so that's directly proportional. The rate of diffusion also become double and an increase. So number 2, the concentration difference has halved. So this is the concentration difference is directly proportional. So if this will decrease, the rate of diffusion will also decrease. So, and the thickness of the membrane has doubled. For example, it is inversely proportional. So if the thickness will double, so rate of the diffusion will increase. So if there's the thickness of the membrane will become double, sorry, so rate of diffusion will decrease. Why? Because this is inversely proportional to the rate of diffusion. So next we have thickness of a membrane has half. So if there's a thickness of membrane reduced to half, and the rate of diffusion will become double. Why? Because it is inversely proportional relationship. So in this way, the option 1 and 4 are correct here. So C is our correct option for this. We're going to proceed to the next question here, and so many calculations are involved in this question. So a student measure the time taken for complete diffusion of a dye into an agar block of a different size, which were suspended in the dye. So we got a different size of the agar block, and we suspend in the dye, and we observe the time, how long it took to complete diffusion of the dye in agar block. So we got the timing, for example, size of the agar block, these are given there. So first size of agar block is a smaller, and then number 2 block, I give the numbers for your understanding. Number 2 block, the size is greater. Number 3 is a further greater in size. And number 4 is a variable size. So what is the predicted time to complete the diffusion of a dye in an agar block measuring the last number 4 block? For example, you can see the number 4 block is 5 into 10 and 15. So what is the predicted rate for this? To understand that, we need to know about the concept. The surface area to the volume ratio is directly proportional to the rate of diffusion. First, by increasing the size, so surface area to the volume ratio decreases. So in this way, it will take more time to diffusion. So that's overall the concept. On the basis of this concept, we're going to solve this question. So first, we'll find the surface area to the volume ratio. So for finding the formula for surface area, for example, first in a question is giving length, width, and the height. So first is the length, and for example, 5 is the length, and 10 is the width, and 15 is the height. So with the help of this formula, we can find the surface area and the volume, the formula length multiply width multiply height. So this will give us the volume. So we're going to find this ratio. For example, if I'm selecting the block number 4, and the surface area of the block number 4 is 550, and volume of a block number 4 is 750. When we're going to divide the surface area to the volume, 550 divided by 750, so answer is coming 0.733. So this is our value. In the same way, I calculated the surface area to the volume ratio of block number 1, and I got the ratio is what? 150 divided by 125, I got 1.2. And in the same way, block number 2, I got 0.6. Block number 3, I got 0.4. And block number 4, I got 0.733. So in this way, if there is a surface area to the volume ratio is 1.2, and in this way, this value of rate of diffusion, what it will take 6.2 seconds. Now, if you're increasing the size, you can see, so if you're increasing the size, the time is increasing. So, and in this way, you can see that the surface area to the volume ratio is decreasing. You can see if the size is increasing from block number 1 to block number 2, and the surface area to the volume ratio reduced from 1.2 to 0.6. So if there's this value reduced, it is taking more time, like 16.1 seconds. And in the same way, if you're increasing further more size, and in this way, the surface area to the volume ratio reduced to 0.4, and it's taking more time. So we are going to predict the block number 4 is after which number. So for example, the time for 0.7 is greater than block number 2. So it means a time will be less than block number 2. And then 0.7 is also less than block number 3. So its time will be less than block number 3. So it means the time of the block number 4 is near to the block number 1. So that's why there is a more similarity that like 0.7 surface area to the volume ratio of the block number 4 is more related to the block number 1. So that's why the correct option or this predicting time. They're not asking about to give an exact time. They're asking to give a predicted time. So this way, the predicted time will be 6.2. Why? Because it is more related to 1.2 surface area to the volume ratio. So here we got idea. So the correct option will be A here. We're going to proceed to the next question. An experiment was carried out to investigate the effect of concentration of sucrose solution on a cell in a plant tissue. So we got a sucrose solution and on the plant tissue, and a sample of the plant tissue were cut into a 7-cylinder of equal length and diameter. So mass of each cylinder was recorded, and each of the 7 cylinders were put into the different sucrose solution concentrations. So we put them in a different sucrose solution concentration, and after 2 hours, the cylinder were removed and bottle dried and then bloated dry and reweighted. So we dry them and then reweighted the percentage of the change in their mass we measured there. So you can see this graph is given there in the question. So according to this graph, we have 3 values are there. For example, one value is above, this is positive value, above the 0. Second value is the same to the 0. And the third value is below the 0. So the value above the 0 is hypotonic environment, mean this is the concentration of the sucrose solution, which is providing hypertonic environment to the cell. So mean the cell has high salt as compared to the outside. So in this way, the water always moves behind the salt. So water is moving from outside to inside. So that's why the mass of these cells is greater as compared to their normal control mass. So in that way, above, from 0 to the above, so to the 20, so that is our hypotonic environment. If you're increasing the concentration of the sucrose from 0 to 0.1, then 0.2, then 0.3, so then rate of inflow of water is decreasing. So that's why the mass is also decreasing. But at the point where it is crossing to this middle line, this is isotonic environment. And this environment, the concentration of the salt inside of the cell is equal to the outside of the cell. So that's why there's no change in the mass of cell. Why? Because the movement of water molecule from outside to inside will be equal to the moving from inside to the outside. What about below the zero? This is hypertonic environment. And in this hypertonic environment, the concentration of sucrose solution is greater outside as compared to the inside of salt. So in this way, the cell will have the high water potential as compared to the outside. The water start moving from inside to outside of the cell, and the mass of cell will decrease. So that's overall concept regarding that. And we are going to understand one more concept regarding this. So number one, we are having a maximum water potential is 0. And that is a distilled water. I mean, if there is the distilled water, mean there is no salt present in the water. So that has a maximum water potential. And this is called 0. And then if you are adding some salt in the water, so in this way, the water potential means concentration of water is decreasing, concentration of salt is increasing. So in this way, the water potential, when it will increase, so this water potential will become 0. That will move to the negative value. Why? Because maximum water potential is 0. So then if you're decreasing it, so this will decrease from 0 to negative value. So water always move. The third concept, the water always move from a high concentration to a low concentration. Mean water is moving from low negative value to the higher negative value. So low negative value mean it is near to the 0, so it has high water potential. And high negative value mean it is away from the 0, so it has low water potential. So water always move from high water potential to low water potential. So that's overall the concept about the water potential. Now we're going to proceed toward the question here. Which row explain the result if the plant tissue cell were put in a sucrose solution with a 0.45 mole per dm cube? So you can see, if you're going to put that on, there's a red spot I put there here. This is 0.45 condition. So this is placed in a hypertonic environment, mean outside the concentration of the salt is more. So water move from inside to the outside, and the cell will have if the water is moving from inside of the cell to the outside. So the water potential of the cell is less negative as compared to the outer environment. That's number one. And number two, the cell size overall, it will decrease. Why? Because water is moving from inside to the outside. So in this way, overall cell mass will decrease. So we're going to see that option. So water potential of the cytoplasm of cell at start of the experiment. When in this type of the question, always focus on what examiner is asking, at the end or at the start. So at the start of the experiment, that's compared with the water potential of a 0.45 mole per dm cube of a sucrose. So cell internal environment at this point has less negative water potential. Less negative mean inside there is more water and less salt as compared to the outside. Outside is more salt. So that's why the water is moving from inside to outside. So what will happen with the volume of the vacuole of the cell? It will decrease. Why? Because the water is moving from inside to outside. So hopefully you understood this question. So A is our correct option here. So we're going to proceed to the next question. The diagram shows the part of the organization of the sections of the DNA molecule and that's associated histone P and R in a prophase of the mitochondria. Sorry, in the prophase of mitosis. So in the prophase of mitosis, what is happening? So condensations of chromatin material starts, number one, and this chromatin material is converting into chromatid, and by making long strings of the DNA with histone protein. So that's overall the concept and what is happening in the meiosis. So you can see that in this diagram. So P, we have a histone protein, R is also histone protein, and Q is a DNA, which is coiling around the histone protein. So there are two styles of coiling. One style of coiling when the cell is present in the form of chromatin material. So in a chromatin material, the coiling is making in the form of cluster to get a close packing in the nucleus of the cell. So, but during the prophase, the cell enter into the karyokinesis. So there's no cell membrane, there's no nucleus right now, and that this is the nucleus has disintegrated. So what is happening? So the DNA is coiling around and it's making strains, long strain. And in this condensation, in the result of long strain, it is producing chromatids. And these chromatids are appearing in the form of chromosome. So you can see like inside of the chromatid, the chromosome, the DNA molecule is condensing and making a long strains. So that's overall the correct concept here. So we're going to see that which option is correct regarding this information. So which statement about the feature of label PQR during the prophase of mitosis is correct? So the quiet DNA molecule form Q and the wrap around the histone R to form small cluster held in a place by the histone protein. It's not going to make a cluster because the cluster will form during the chromatin formations when the cell is not dividing. So that's not describing the condensation lead to the chromatin formation. So that's why A option is wrong here. Now the B option, the group of histone protein P and its associated DNA molecule and Q move closer together as the chromosome condenses around R. So in this way, if there's a histone protein, that they are coming closer. So, but here they are not describing about the chromatid formations. So in this way, that's all these the histone protein and their associated the DNA molecule. So they are not condensing to make strain and to produce the chromatid. That's not describing. What about chromosome condenses around the around the R group? It is the wrong statement. So chromosome is not condensing. It's the DNA molecule. So that's why it is a wrong statement. Second, the C is here. The histone P and R are made up of the protein around with the DNA molecule. So Q is wrapped so that the DNA molecule can fit into a nucleus. So you can see there's no nucleus in a prophase of mitosis. A nucleus has been disintegrated. So that's why it is a wrong option. So the link of the group of histone protein, P and R, and associated DNA molecule form the strand that fold twisted together and form chromatid. So this is exactly describing what is happening in the prophase of the mitosis. So first they will quite around each other and produce a long strains, and these long strains are making chromatid of chromosome. So D is our correct option here. We're going to proceed to the next question. How many copies of each DNA molecule would be found in the cell at the start of the stages of mitosis in a cell shown there. So the cell mitosis is consist of what? At the stage of the mitosis, the cell is consist of different phases. So for example, G1 is a part of interphase. And cytokinesis is the last phase of M phase. So M phase means when the cell is dividing. So we're going to see that what is happening at this point. For example, at the start of the cytokinesis, cell contain two sets of the chromosome with single chromatid. So each set is present on one pole of cell. For example, the chromatid got separated at anaphase, and in the telophase, they are moving toward the opposite. They reach to the opposite pole, and then the cell is dividing. So this is cytokinesis has taken place. So in this way, overall, in the cytokinesis, until now, the cell did not complete. Until now, the cell didn't complete the cytokinesis because the question is start. So at the start, the cell is in the form of the one entity. So in this way, within the one boundary of the cell membrane, there are the chromatid are present. So that's why there is the two. What about the G1 phase? After the cell division, it will enter into the growth 1 phase. And in the growth 1 phase, obviously, each newly dividing cell will receive one chromatid. So if there's a one chromatid is there, so within one chromatid, there is a one DNA molecule. So in this way, there will be one DNA molecule in the G1 of the interphase, and there will be two DNA molecule in a cytokinesis at the start of cytokinesis. So here, examiner can change a bit. For example, if it's written there at the end of these phases. So when we talk about at the end, so then option will change for the cytokinesis. Why? Because at the end of the cytokinesis, one cell will divide into two, and in this way, condition will be different. But most probably, the question may come related to at the start of these stages. So option B is correct here. We're going to proceed to the next question. One characteristic of DNA is that in a universal genetic codon. So what is universal genetic codon? So what's meant by universal genetic codon? So all living organisms use same triplet code for amino acids. So we have 20 different type of amino acid, and these 20 different type of amino acid which are naturally present throughout the living organism. So whatever the living organism they are having protein, they composed of the naturally existing 20 different type of amino acid. And these amino acids are produced by the same code of triplet codon in all living organisms. So that's a concept of universal genetic code. So A is our correct option here. And we're going to see that the option B, all DNA triplet code for the different amino acid. So that's not correct regarding the universal code. Why? Because they are giving different organisms, the different triplet code are producing different amino acids. So that's not correct. So not correct regarding universal codon, and it's against the concept of universal codon. Okay. Now, step number 3, not all DNA triplet code for a amino acid. So that's again the concept. It's not coherent regarding the universal concept. Mean the old DNA triplet code for not for an amino acid. So it's against the concept of the universal genetic codon. While all the living organisms contain same 4 nucleic acid. What about here? Supposed to be a nucleotide. For example, that might be a correct option. But another way, there's a nucleotide are adenine, guanine, cytosine, and thymine. What about thymine has been replaced to uracil in messenger RNA? So what about here? The word nucleic acid is wrong. So all the organisms, they are having same code or same nucleotide. So these are given there. So that's why this option is wrong here. So correct option with the correct definitions of the universal genetic code, that is A. Which statement about the messenger RNA is correct? So it is the polymer made of nucleotide all joined with hydrogen bond. So that is the wrong option. Why? Because these polymers are made up of the different nucleotide. They joined by phosphodiester bond. So that's wrong option. Second, each nucleotide subunit contain sugar ribose. So each nucleotide that contains sugar ribose, that is the correct option, because every nucleotide contains one phosphate group, one sugar ribose sugar, there is 5 carbon sugar, and one nitrogenous base. So that's the correct statement. The option number C, it always has an equal proportion of adenine and uracil. So a proportion for adenine and uracil is not equal. Why? Because that's not completely the double-stranded molecule. So that's why the options for adenine is coming opposite to the uracil. It's not equal in amount. So second, on the D, the messenger RNA sequence in identical to the template strain of the DNA. So whatever the template strain sequence in the DNA, the messenger RNA is complementary, or you can say it's opposite to nucleotide that's present in the template strain in the DNA. For example, if there's a template strain in the DNA, there is a cytosine, so opposite will be guanine on messenger RNA. So that's why it's a wrong statement. So our B is our correct statement here. We're going to see here, we have the diagram show the part of the process of translations in messenger RNA. So this messenger RNA is combining with transfer RNA. So you can see that's when it's combining with a transfer RNA. So that's the part X is our anticodon. And the part Y is a codon, and the codon is making hydrogen bond with the anticodon. And second, what about Z is overall is a transfer RNA. So what are the name of the structure label X, Y, and Z? So we're going to see that what are the correct labeling according to that. So X is our anticodon, Y is codon, and Z is transfer RNA. So B is our correct option here. We're going to proceed to the next question. The DNA triplet of the genes are translated as amino acid to stop signal during the protein synthesis. So the tables show some of these triplets are given there. So here we can see the DNA triplets are given in the one column. What about the name of amino acid that produced in this result that are in second column? So here we have a stop signal that's ATC. These are given there. What about some other amino acids are given there? So what would be effect of one substitution mutation in a triplet coding for tyrosine? So for example, the tyrosine is coded by ATA and ATG. So if you're replacing one nucleotide from there, so what it will produce? What is existing in the table? So for example, if you're replacing ATA and we are replacing this thymine in the center to cytosine, so that will produce ACA. And you can see ACA is what is present. So ACA is producing cysteine. So it means it is correct statement. So the triplet is translated as cysteine. So it's possible, for example, if you're doing one replacement of T into C, so that will produce cysteine. So this is correct statement. The second statement, the triplet is translated as tryptophan. So tryptophan means this is ACC. So if you have a choice, for example, to replace ATA to anyone to produce ACC, that's not possible. In the same way, ATG to replace anyone to produce ACC, it's not possible. So that's why it will not produce tryptophan, because there is more than one nucleotide difference. Second, we have the triplet is translated as the tyrosine. So this triplet can be translated as tyrosine. Why? Because ATA, if you're replacing only the G here, A to G, so that will produce. So that's why this is possible. What about translation stop at this triplet codon? So we're going to see that the stop signal is ATC. So the translation will stop. So this is our mutant triplet code. So if you're replacing, for example, you are having A and T and C. So if there's a one replacement between that, for example, if your replacement of one part, so you are doing replacement of ATA to ATC. That one. You're moving from ATA to ATC. There's one replacement, and this replacement will produce a stop codon. And this stop codon will produce what? A stop signal, and in this way, the translation will stop in this point. So our correct option are included. That's our following point. So 1 is correct, and 3 and 4 are correct. So C is our correct option for these questions. So we're going to proceed to the next question here. Which row identify the cell with plasmodesmata? So plasmatasmata is a cytoplasmic connections between the two living cell. So for example, we're going to take one example of cytoplasmic connection cell. So you can see one is in a xylem, we have companion cell and the sieve tube element. So there is a connection between companion cell and a sieve tube element. This cytoplasmic connection is plasmatasmata. So that's present. The phloem sieve tube element, this is one that contain plasmatasmata. What about we have companion cell, this contain plasmatasmata. What about in the right side, you can see here the xylem. So xylems are dead cell. And in these dead cell, there's a thin cell wall present between two adjacent cell, and they're making what? Pit area. This is not cytoplasmic because there's no cytoplasm in the xylem because these are dead cell. So that's why this will be a wrong option here. So correct option here with us is B is our correct option. So phloem sieve tube element have that. Companion cell have that. And the xylem vessels element, they do not have plasmatasmata. So we're going to proceed to the next question. Which statement about the apoplastic and symplastic pathway are correct? So apoplastic pathway is what? The movement of the water through the cell wall. So, and the cell wall to cell wall, for example, these are two adjacent cells are there, and these adjacent cell, you can see the black color of arrow is moving only in the cell wall. That is what apoplastic pathway. But when the water will reach to the endodermis of the plant of root, and inside of the endodermis, we have Casparian strap. There is a lipids there, and the water has to move into the cell to pass into the endodermis of Casparian strap. So that's why this is a red color of arrow showing symplastic pathway. So symplastic pathway mean the water is moving from cell cytoplasms to cell cytoplasm. So we're going to see that which option is correct for both. So if the apoplastic pathway, in the apoplastic pathway, water move through the free spaces in a cellulose cell wall of the plant root. So that's a correct option. So water is moving in the free spaces between the cellulose and in the number 2, in the symplastic pathway, the water molecule diffuse through the cytoplasm. So the water is moving inside of the cytoplasm. So that's why it is correct option. Number 3, the water molecule traveling through the plant tissue moved by mass flow along the apoplastic pathway. So water is moving along apoplastic pathway with dissolved salts. So that's moving from a high water potentials to the low water potential. So that's a mass flow method. So this is our correct option here. What about Casparian strip block the symplastic pathway and forces all water to move through the cytoplasm of the endodermis. So Casparian strip is basically they're blocking apoplastic pathway, not symplastic pathway. So that's why the water changed the path from apoplastic pathway to symplastic pathway. So that's why this is wrong option. Our correct option is that's a B, which contain 1, 2, and 3 are there. So here we're going to proceed to the next question. Where does the water evaporate during the transpirations? So during the transpirations, where the water is evaporating. So if the water is moving from the one cell to another cell, it is crossing the boundary or cell membrane. So this is osmosis. But when water reached the side of water move from cytoplasms to outside of the cell on the cell wall. So if the water is moving from cell wall to the spongy mesophyll free spaces, this movement is that is evaporations. So in that way, that's called evaporation. If the water moves from spongy mesophyll free spaces to outside of the leaf, this is called transpirations. So in that way, according to the terminology, the evaporation, so the correct option will be what? The mesophyll cell wall of the cell. So if the water is evaporating from the cell surface of a mesophyll cell wall, so this is evaporations. What about the water is moving intracellular spaces? This is our diffusion. So next second is the leaf surface. So that's not include that part of the evaporation, the terminology what we use within the it's not part of the transpirations. So second, a stomatal pore. This is called transpirations by diffusion process. So correct is what? If the water is evaporating from the cell surface, so within the cell, it is the part of transpiration, and this part of transpiration is called evaporations. So that's why the C is our correct option. We're going to proceed to the next question here. And the diagram show the outline of 3 xerophytic leaf of the same type in 3 different conditions, P, R, and S. You can see in a condition P, what is happening? The water is moving from outside to the inside of the cell. That's why the leaf cells are turgid. It's visible. They aren't turgid. Why? Because water moved from outside to the inside. So what does it mean? It means the water is, this leaf is present in dilute solution, and the salt concentration outside is less. So that is hypotonic environment. While the cell P and S and the water move out from inside to the outside, that's why this is shrinking. These leaves are shrinking. So in that way, this means the water move from inside to the outside. So that's why this is a shrinking. So it means they are present in concentrated solution. What about the level of concentration is different in R and S? So you can see R is present in more concentrated. So that's why it is more plasmolized. What about the S is present in less concentrated solution? So it is less plasmolized. So we can have, like, for example, the water potential. So we can compare with the reference of water potential of these results. So which descriptions of the water potential of cell in the layer Y is correct? So water potential of the cell in the layer Y. So you can see the water potential in a cell in a P is a higher than R and S. So why? Because the water moved from outside to the inside of the cell, and inside of the cell, there's a more concentration of the water and less salt as compared to the R and S. So that's why the water potential in the P cell, why? Because water moved from outside to the inside, it's a higher as compared to the R and S. Now, next is comparison between S and R. So if we talk about R, so it has lower than P and S. What about the S, the water potential, because it lost less water as compared to Y, and it has less water. So that's our, sorry, that's Y is lost more water, R is lost more water, and S lost less water. So that's why the water potential of S is lower than P only. So what about its water potential is greater than R? So that's why this is, you need to understand in this way. So what about the P is present in dilute concentration, dilute solution, the R and S are present in concentrated solution, but the concentration of the R is more as compared to the S. So that's why the R is losing more water. So that's why it's the water potential in the cell of R is lower than P and lower than S. What about in S? So water potential in S is only lower than P. What about it has a greater than R? So that's why A is our correct option here. So I have explained regarding the osmosis phenomena, how it's working in the previous question. So hopefully you will understand that. I'm going to proceed to the next question here. So the next question is also related with a xerophytic plant. The photograph show the transverse sections of the leaf of species of a grass. So that's the leaf of species of the grass. So you can see this is part of marram grass, and this marram grass is behaving at the daytime. It rolled up its leaf to reduce the surface area for conservation of water, because there's no direct sunlight is coming, and it's coming on the surface of this leaf. Second, it has sunken stomata, I mean hidden stomata inside. And the third, it has trichomes, or it has the hairs on the leaf surface, so which entrap the water vapor, and that's why there's a high water vapor inside of this the leaf, so that increase humidity. So if there's a high humidity, so in this way, the low rate of transpirations. So if there's a low rate of transpiration, so in that way, that will be conservation of water. So leaf roll up and trap the water vapor inside of the surface and the sunken stomata. So that's why it decreased the transpirations of water. That's a strategy for decreasing the transpiration of water. So we're going to see that the question, the grass is specially adapted to grow in a dry habitat, which will correctly explain how the feature helped the grass to grow in this habitat. So hair-like structure, these are given there inside, and the leaf shape. So that's one. So what is a hair-like structure is that the hair-like structure increase the internal humidity. Fine, it's correct. What about and the leaf shape decrease external humidity? It's also increasing humidity. Why? You can see it's a ball like structure, and inside that's making curve shape, and this is entrapping the water. So that's why this option will be wrong here. So this decrease the humidity. That's wrong. So what about it decrease external humidity? So leaf are hair-like structures are entrapping the water vapor, so that's why it is increasing. So humidity, so that's why it is the wrong here. So what about the C is option? It increase the internal humidity, and the leaf is also increase the internal humidity. What about in the D option? That's a decrease is a wrong here, and decrease is a wrong here is option. So here, the C is our correct option here. We're going to proceed to the next question. Which statement supports the theory of active loading of sucrose into a companion cell? So during the loading of sucrose, during the loading phase of a sucrose into the companion cell, so what is happening? So the pH decreases in the cell wall of the companion cell as compared with the cytoplasm. So that's the correct option. And to explain that, I have mechanism. So we are going to proceed to that mechanism, how it is working. So you can see we have a left side, I have the phloem, whole phloem tissue, and this phloem tissue is contained, the three cells are there. Now, number one cell, we have a phloem, the sieve tube element. This is a phloem cell. And this phloem cell, beside the phloem cell, I have a companion cell. And beside the companion cell, there's a photosynthesizing cell is present. So what is the loading? In the loading process, the sucrose transfer from photosynthesizing cell into the companion cell, and then it transfer from companion cell into the phloem cell. So the hydrogen ion are pumped into the cell wall, which is present between photosynthesizing cell and companion cell. So when these hydrogen ions are pumped out, they're not going into the cell. They are staying between the cell wall. They are present inside of the cell wall. When the concentration of this hydrogen ion increase between the cell wall, so in this way, they start moving from outside to inside of companion cell. And when they start moving by facilitated diffusion, so the protein channel will open for them. So same protein channel will be used by the sucrose to move from photosynthesizing cell to the companion cell. And this is called co-transport. Now, when the concentration of the sucrose will increase in a companion cell, so this will move from companion cell to the sieve tube element by plasmodesmata. And in this way, the loading will take place. Now here there's a question, during the loading, what is happening where the pH value is decreasing? So the pH value, we defined a negative log of concentration of hydrogen ion in a solution. So the concentration of the hydrogen ion is increasing in the cell wall, which is present between the photosynthesizing cell, or you can say a source cell, and companion cell. So the pH value is decreasing in this cell wall in this region. So that's why to understand that, we can understand this whole phenomena. So the correct option will be the pH value is decreasing in the cell wall. So we are going to proceed to the question here. So option is here, which statement support the theory of active loading of a sucrose? Active loading, why? Because the ATP is involved in this loading process. So that's why it is called active loading. So The A option, the pH value decrease in a cell wall of a companion cell as compared with the cytoplasm. So that's our correct option. What about the pH decreases in the cytoplasm? So pH not decreasing in a cytoplasm of companion cell. Why? Because it's not increasing hydrogen ion concentration. So pH decrease in a companion cell, it's not, it's decreasing in a cell wall, and a pH decrease in a sieve tube element, the hydrogen ion are not going in a sieve tube element, so that's why it will not, that's our wrong option. So A is our correct option with the given diagram as I explained. I'm going to proceed to the next question. Which property of water are essential for its role to transport of blood in a mammal? So what is the property of the water as a transport agent or material or transport medium? So it has a high latent heat of vaporization. So high latent heat of vaporizations mean it's absorbing more heat, and when it's evaporating, it's absorbing more heat. And this is the property of water molecule. So that's why the water molecule is used to balance the temperature of the plant and an animal. So that's why the animals, they are releasing sweat on their surface and they are evaporating it. What about the plant? They are performing transpiration, and in the result of transpirations, they are maintaining their body temperature. This question is not regarding this. This question is asking about what is essential for transport of the blood. So that's why it is wrong here. Solvent for polar substance. So all polar molecules are soluble in the blood. So that's why these all polar molecules, they are moving from in the dissolved form from their source to the place where they're being consumed. So that's why water that's present in the blood, and that's also a solvent. So this is giving a solvent property to the blood for transport of the material. So B is our correct answer here. We're going to proceed to the next question, and this also related with the red blood cell. So the diagram show some of the events that happen between the plasma and the red blood cell. So for example, if the red blood cell is present near the cell of a body cell, which is performing that's aerobic respiration, and the cell is producing carbon dioxide, and this carbon dioxide move into the plasma, and this plasma fluid that's moved from a high concentrations to the low concentration inside of the red blood cell, and this carbon dioxide will combine with the water. So in the presence of carbonic anhydrase enzyme, and that will produce the hydrogen carbonate. Now, this hydrogen carbonate is not a stable molecule, and this will dissociate, or this will split into its ion in the form of the bicarbonate ions, and these hydrogen carbonate ion will move from inside of the cell to outside. So what about a buffering effect? So in this way, the chlorine ion negative, that will move from outside to the inside. So this is not buffering. This is basically balancing the charges across the membrane of red blood cells. For example, one negative charge is moving out. In the same way, the one chlorine is moving from outside to inside. So that's a balancing of charge across the cell membrane of red blood cell. Second, hydrogen ion, that's number 3, is a hydrogen ion. So these hydrogen ion will combine with the hemoglobin and produce hemoglobinic acid. And in this way, the affinity of a hemoglobin with oxygen will decrease, and it will unload oxygen, and that will proceed to the plasma cell. So that's overall the process, what is happening in the body tissue with the red blood cell. We're going to proceed here. So what is the question? In a question, what do the number 1, 2, 3, and 4 represent? So you can see number 1 is what is happening. So number 1 is our carbon dioxide. And number 2, these are hydrogen carbonate ions. And number 3 is our hydrogen ion. And number 4, so that's our chlorine shift. So this chlorine shift is moving from outside to the inside. So to balance the charges across the cell membrane of the red blood cell and blood. So that's a B is our correct option here. We're going to proceed to the next question. The diagrams show the walls are in the heart when it's viewed in a cross-sections from above at the different stages in a cardiac cycle. So if you see that in a first diagram number 1, so semilunar wall is closed. What about atrioventricular wall is open? So it means the blood is moving from atrium to ventricle. So here there are two possibilities there. One possibility is that there is atrioventricular diastole, mean atria and ventricle both are relaxing and the blood is moving from atrium to the ventricle, and a semilunar wall are closed. Why? Because there's a high pressure in aorta and pulmonary artery. So second possibility is that there is atrial systole. The atrial systole mean there is a high atrial pressure and the blood is moving from atria to the ventricle. So that's why atrioventricular wall is open. There's the two possibilities are there. What about in the diagram 2? So you can see that atrioventricular wall are closed. What about the semilunar wall are open? So if the semilunar wall are open, so what does it mean? There is only one possibility, ventricular systole. So if there is a high pressure in ventricle, so the blood is moving from ventricle to outside of the heart in aorta and pulmonary artery. So that's why it's only the possibility. So we're going to check the option related to that. So during exams, you can write these options with the help of your pencil, so that will be helpful for you. Which stage is in a cardiac cycle is shown there. So in the same way, in the diagram 1 is a diastole and diagram 2 is a ventricular systole. So that's a correct option. So we're going to see that in a B, the atrial systole, it is correct. What about in a diagram number 2? So you can see it's a diastole is wrong. What about in C? In the diagram 2, diastole is wrong. Why? Because it is confirmed this is ventricular systole. And in the D, is the atrial systole. It's also wrong. Why? Because it's confirmed it is ventricular systole. So this way, A is our correct option here. We're going to proceed to the question number 35. So where are the squamous epithelium cell found in a human gaseous exchange system? So you can see in a gaseous exchange system on the surface, so squamous epithelium cell, they are present in alveoli. So that's the alveoli, that's our squamous epithelium cell that only present there. What about the trachea and bronchus? So these trachea and bronchus, they contain columnar epithelium cell with a goblet cell. They are in between. So these columnar epithelium cell, that's normal structure that present in trachea and bronchus. What about the alveolus? So this contains especially the squamous epithelium cell. So that's our correct option. So D is here. So why? Because it's only take for the alveolus here. Now, question number 36, which statement about the gaseous exchange between the air in alveoli and the blood in pulmonary capillary is correct? So this question is, you need to falsify every statement to find the correct statement here. So the oxygen concentration in a capillary leaving pulmonary artery is higher than the oxygen concentration in alveoli. So if there's a word, higher is wrong. So because the pulmonary capillaries are getting blood from the pulmonary artery. So in this way, the blood from pulmonary artery coming from the right ventricle of the heart. So if there's the blood is coming from right ventricle of the heart to the pulmonary artery, and then pulmonary artery that's moving to that's our pulmonary capillaries, so it's leaving the pulmonary artery. So in this way, they contain deoxygenated blood. So this deoxygenated blood has low concentration of oxygen. So that's why this option is wrong here. So number B, the gases must diffuse across endothelium of pulmonary capillary and the endothelium of alveoli. So the correct option will be what? The gases must across or diffuse from endothelium of pulmonary capillary. So, for example, high concentration of the carbon dioxide is there. That will move from endothelium of pulmonary capillary, and this will absorb on epithelium of alveoli. So this epithelium cell on alveoli, they're increasing the surface area of the alveoli. So the gases are exchanged there from the surface of large surface area, which is created by epithelium cell. So that's why that will be a correct statement. So endothelium is a wrong statement here. So C, the elastic fiber in alveoli wall allow the alveoli to expand to increase the surface area during the pulmonary, it's available for the diffusion of pulmonary capillaries. So that's a correct statement in a sense. Why? Because the elastic fiber, that's our fibrous protein. It has a high elasticity, and they're present in the wall of alveoli. So when we inhale, so they expand to increase the surface area for a gaseous exchange, and during exhalation, they recoil and help in exhalation process. So that's why the C is our correct option, while the D, the breathing out, reduce the carbon dioxide concentration gradient between the blood in the pulmonary capillary and in the air and alveoli. So when we breathe out, so that this will transfer. But why? Because the 4% of the carbon dioxide is present in the lungs. So during breathing out, this carbon dioxide concentration move from inside to the outside, and this will increase the difference of concentration gradient. Why? Because the concentration gradient in the blood is 4, and then in the lungs, this will further, that will decrease when the carbon dioxide will move from inside to the outside. So that's why the word reduce is a wrong. Here it's supposed to be increase. So C option is correct here. We're going to proceed to the next question. The average thickness for some structure in a human respiratory systems are given there. For example, human cell membrane is given there. That's a 5 nanometer. Cytoplasm of alveolar cell wall is 190 nanometer, and cytoplasm of capillary cell wall cells are 90, and a tissue between the alveolar wall and capillary wall is 300. Now, these different options are given there. So we are going to proceed to the answer. A molecule of oxygen in an alveolar space next to the wall of the alveoli. So that's moving from alveolar space to next to the wall of alveoli, and then it's moving toward the red blood cell. So what is the shortest distance that the molecule need to diffuse from its correct position when it's present inside of the alveoli to the hemoglobin, which is present in the red blood cell? And we consider like there's no distance between red blood cell and the capillary wall. So we need to calculate first, we need to consider the red blood cell has to pass 5 cell surface membrane if it's moving from alveolar space to the red blood cell. So for example, this red blood cell is, this oxygen is first, it will enter into the alveolar cell membrane, and then it will go out to the alveolar cell. And that once again, it will enter into the capillary cell membrane, and then it will go out from the capillary cell. These are 4. And then it will enter into the red blood cell. This is 5. So it has to cross 5 cell surface membrane. So that's why we're going to add this all distances given there. Just we are going to multiply 5 multiply 5, because cell membrane will be 5 times and plus 190. That's the alveolar cytoplasm, and plus 90. This is our capillary cytoplasm. Well, the 300. This is the tissue between alveoli wall and a capillary. So overall distance will become 605 nanometer. So this type of calculation, it's a simple calculation, just we need to manage how many number of the cell membranes are there in between. So here, the C is our correct answer here. Which statement refers to the disease as tuberculosis? So which statement explain why the antibiotic treatment for the tuberculosis takes a longer time? So when we talk about antibiotic treatment, so this is a artificial immunity. So we are generating artificial treatment. Artificial mean antibiotic which kill the bacteria, and they're killing bacteria by stop making their cell wall, and they're stopping the cell to divide, and there's the mechanism. So in this way, they are killing or stopping the growth of the bacteria. So in this way, which option is correct regarding which is delaying the treatment of tuberculosis? So option number one, the pathogen is not accessible to immune system. So this immune system is not involved in the treatment of antibiotic treatment. Why? Because this is our natural immunity. So immune system mean our phagocytes and our white blood cell, B cells. So and the T cell, they have access. So in this way, there's not relevant to the antibiotic treatment. So option one is wrong here. So bacterial pathogen reproduces slowly. So that's why because if there's a bacterial pathogen and reproduce slow rate, so that's why its effect on formation of the cell wall is less, so that will delay this. So it will take a long time. Second, the pathogen is not very sensitive to the antibiotic. So this way, if the sensitivity of pathogen is less to the antibiotic, so the effect of antibiotic will be less on the pathogen. And in this way, that will take a longer time of this treatment. So option number 2 and 3 are correct. So D is our correct option here. We're going to proceed to the second last question here. The average size of some pathogen are shown. So these pathogen are what? So we have HIV particle, their size is given there. Mycobacterium tuberculosis for TB, their size is given there. And Plasmodium and Vibrio cholera. So that's our, so we're going to see what is the question. The one type of air filter has been shown to be effective at preventing any pathogen of 1 micrometer or larger from entering to the air system of a room. So based on size, number one criteria, and mode of transmissions, which disease would air filter prevent the entering of air systems of the room? So you can see on the basis of size, we are going to size and mode of transmissions. So first, the air filter can filter those type of the diseases which are spreading by air. So that's number one. And number two, those types of diseases which size should be less than 1,000 nanometers. So you can see, if there's a more than 1,000 nanometer, that's is equal to the 1 micrometer. Or if there's a more than 1,000, that can easily be prevented. So there are two conditions. Number one, it's supposed to be airborne disease, or it's air spreading disease. And number two, size should be more than 1,000 nanometer. So on the basis of this criteria, only one disease is coming, Mycobacterium tuberculosis. This is airborne disease. This is spreading by contaminated air, number one, and its size is 300 nanometer. So it means it is equal to the 3 micrometer. So that's why this will be prevented by the air filter. Rest of other disease, HIV is not an airborne disease. It will directly spreading from one body contact to another body contact by a body fluid like blood transfusions. In the same way, Plasmodium is by vector or is by mosquito. It transferred by mosquito, so that's why it will be not prevented by air filter. And Vibrio cholera it's spreading by contaminated water. So that's why air filter will not work it to prevent. So that's why the correct option will be D here. So only tuberculosis. Why? Because due to its size and due to its mode of transmission by air. We're going to proceed to the next question. A successful vaccinations program provides a level of immunity where the majority of the population is protected. So there are several factors that can affect the success of vaccination program. So which row correctly show the factor that can affect the success of vaccination program? So here, you need to see that which factor are affecting or improving the successful vaccination program. For example, frequent mutation of pathogen. So if there's a pathogen is frequently mutating, so in this result, the pathogen sensitivity become less to the antibiotic. So this way, that in that condition, this is also involved. It's affecting the vaccinations program. So for example, if you do the vaccination of one type of pathogen and it got a type 2, for example, what is happening with the coronavirus, so that's getting different variant. In the same way, it's happened with the AIDS. So if you are producing one type of the vaccine, if it's more successful and it has frequent mutations of another strain, so that create a problem. So vaccine become ineffective regarding that. So that's one. This option is correct. Vaccinations from 8 weeks. So the time is very less. So you can say like vaccine can stay effective until 24 months with the proper storage conditions. So that's why this is a wrong option. It's not affecting. Pathogen is able to invade in a T cell. This is called pathogen concealment. So the pathogen can hide itself inside of the T cell or this white blood cell. So that's why the vaccine cannot work inside of the cells or pathogen which is present inside of our immunity cell. Why? Because when the immunity cell will become stronger, but it can kill others, it cannot kill itself. So that's why that's called pathogen concealment. Second, we have a booster vaccination need frequently. So certain type of the vaccines which need a booster. Why? Because after getting second dose, still the patient is affected by same pathogen. So it means this vaccine is not triggering immunity system to produce enough the memory cell, and which can produce enough the antibodies. So that's why we need a more booster for that. It happens regarding the COVID, the vaccine. So some strain of the COVID, some type of the COVID vaccine, we need a third dose. That's a booster dose. So if we need a frequently more booster dose, it is affecting the vaccination success of vaccination program. So that's why B is our correct option here. So that's why all are correct. Only the time 8 week, the vaccine, if there's a proper storage conditions are there, the vaccine will stay normal. So I think I have taken a bit more time because a certain type of questions were there. They need more explanations regarding that. So hopefully it will be helpful for you. If you find any problem related to any question, so you can also put your question in the comment box. I'll try to answer that question. Thank you. Thank you very much.